So what are the differences between microarray-based technology and sequencing technology? So the first one is their principle. Microarray-based technology is based on hybridization between the probes and the sample cDNA, while Sanger sequencing is based on high-throughput sequencing. So the background noise, it is higher for microarray because they will tend to have cross-hybridization, while sequencing technologies tends to have lower background noise compared to microarray. For their dynamic range, microarray dynamic range is lower. It can go up to a few hundredfold compared to sequencing technology, which has a higher dynamic range that can be more than 8,000-fold. The reliance on genomic sequence. So microarray will need to depend on existing knowledge of the genome sequence, while sequencing would not need to rely on the genomic sequence. Their ability to distinguish different isoforms. So microarray, there's a limited potential in distinguishing or identifying isoforms, while sequencing, they will have the ability to differentiate. So for the next generation sequencing, these new technologies constitute various strategies that relies on a combination of template preparation and sequencing technology. So we'll first compare the differences between first generation sequencing, Sanger sequencing, and next generation sequencing. So in both cases, you will need to fragment the sample DNA. So both of them are fragmented into smaller sizes. And for Sanger sequencing, it needs to be ligated with the plasmids and transformed into the E. coli. So once it is being transformed, it will need to go through the cycle sequencing where each of the reaction will incorporate a labeled dideoxynucleotide, fluorescently labeled ddNTPs. So from this cycle sequencing, you would be able to determine each individually fluorescently labeled nucleotide and determine their sequence through the electrophoresis and the laser excitation. So for next generation sequencing, they have the same methods of fragmenting the DNA, but they do not transform into bacterial libraries, but instead they will tend to ligate with adapters. So the dark blue color here is the unknown DNA fragment, while the adapters are known sequences. So all of them are ligated with adapters, and they will have multiple copies of the fragment DNA being attached or anchored onto a surface. Okay, and so from here, they will be able to perform their cycle sequencing. So for example, there are 1, 2, 3, 4, 5, 6, 7, 8, 9 locations of the DNA fragments in this region. So it is multiple or parallel sequencing of all these DNA fragments at the same time, which is different from capillary electrophoresis, where they only have one read per capillary, in contrast to more than 10 to the power of 6 reads per array that can be done. So you can see that there's a higher throughput for next generation sequencing. So for each individual location, for example, for this location, the first base would be A. What is base 1? The first location here would be A, and the next nucleotide would be G, and the next nucleotide would be T. So for this fragment, the sequence would go like A, G, T. For this position, it would be C, T, T. Okay, so I will continue with the next slide to look into what are the different platforms for next generation sequencing. So we'll go through one platform by one platform. We'll start off by going into 454, or the platform developed by Roche. So the 454 system was the first next generation sequencing platform available as a commercial product. So they generate their templates by using emulsion PCR. Okay, so we look into how does emulsion PCR works. So emulsion PCR is a reaction mixture consisting of an oil-aqueous emulsion to encapsulate each of the beads. So in any one droplet, oil-aqueous droplet, they will have one bead with one template. So what will be happening in this oil-aqueous droplet is that PCR amplification would be performed within these droplets. So as you can see here, all the templates are being ligated with adapters, and these adapters will be able to bind to the sequences on the beads. So from here, they would be able to perform PCR amplification. So once PCR amplification is done, okay, as you can see here, there will be dissociated template dissociation. And once the PCR amplification is done for a number of cycles, you can see from the original one, which is a bead with only the adapters. At the end of the reaction, it would be the bead with the amplified fragments. So what happens to these beads is that these emulsion PCR beads will be deposited into a pico-titer plate wells. So it will be deposited in a multiple well platform where each individual well will be having one bead. And they will be covered, each of the bead will be covered by the sulfurylase and luciferase beads. Okay, they are covered by multiple beads with sulfurylase and luciferase. So how do they sequence, or know what are the sequences contained within this fragment? The blue color is known, the adapter for blue colored region is known, and red colored region is also known. The unknown part is over here, right? So the red region is unknown, the red region is known, here is the unknown, and this is the known region. So they will attach with a primer and perform the polymerization reaction. So let's say in any one reactions, they will deposit with C nucleotide. If let's say the template consists of a G, the C nucleotide would be able to bind here and the reaction will occur. Okay, and the luciferase will release light and it will be detected by the detector and relayed to the computer output. So if let's say this is not G, it is A, then this would not extend. So this will stop here. So you can imagine that with all the beads in different wells, some of the beads will go faster, some of them will go slower, depending on the incorporation of the nucleotide. Once the nucleotide is being incorporated, the light will be released and being recorded. So during the sequencing process, light emitted from the phosphate molecule during the nucleotide incorporation will be recorded. So what are the limitations of this 454 technology? So let's say you have G in your template. So your sequence here would be C, C, and C, right? So a limitation for this 454 technology is that they may have homopolymers, for example, AAA or GGG. Because there is no terminating moiety, the reaction will not stop. So at any given cycle, the length of all homopolymers must be inferred from the signal intensity. So for example, some of the strands would go faster and some of the strands would go slower. In any cases, if you have multiple nucleotides which are the same GGG or AAA, some of them may be added additionally or it may be misincorporated. So at any one point, the error type would be more nucleotide being inserted or some of the nucleotide are not inserted, so it results in a deletion. So the dominant error type for the 454 platform is insertion deletion, due to the incorporation versus non-incorporation. So sometimes there may be more A being incorporated, or sometimes there may be less. It's because there is no stopping for this reaction, no terminating moiety. So the dominant error type for this 454 platform is insertion deletion. However, what are the key advantages for 454 platform is that they have a long read length. So they can read up to 200 and 300 base pairs, which is the longest read length among all the NGS platforms. So for this platform with long read lengths, they are suitable for applications like de novo assembly and metagenomics. So we'll look into some of the examples of the 454 sequencing platform, which is this video.